Beta sheets are formed between, we have some protein strands of our protein now. Our chain is folding, kind of. We can either have these adjacent strands oriented parallel, so all of our N termini are on one side and all of the C termini are on the other side, or anti-parallel, right? So we have N to C and then C to N. What's sort of cut off in this diagram in your book is sort of what loop sections would need to connect these, right? These are not separate proteins interacting. This is sort of one continuous chain folding back on itself. The H bonds here are shown as these sort of aqua colored dots. Are the side groups interacting or is the backbone interacting here in these H bonds? Side groups are the orange circles, right? Whatever that might be. It's still the backbone. Alpha helices and beta sheets form to satisfy backbone H bonding, right? We don't know what the side groups are, so they may or may not be able to participate, okay? And the only difference here is, of course, it's no longer this every fifth one is involved. It sort of depends on how long these folds might be. But again, the backbone's H bonding still with itself. This chain is turning somewhere up here and has these unstructured, maybe loop regions at each end. So this is, again, just kind of a nice summary slide for you. Yes? No, it doesn't. It can be a mix. It's just a lot less likely in nature. Typically, it's going to be all oriented parallel like this, or it's going to alternate. But you could have a mix that's like N, N, and C. It's just rarer. It's just less likely. But it's possible. It's definitely possible. These are just much more common. So again, this is a summary slide, talks about the difference in length, and then talks about, again, alpha helices satisfying H bonding every fifth amino acid, or beta sheets satisfying H bonds between strands. Again, this is all one protein. Remember, we're still talking about one continuous thing, but it's just sort of adjacent strands. All right, so we have a summary visual of primary structure from N-terminal to C-terminal. The order of amino acids is primary structure. Whether that primary structure can form into alpha helices or whether it can form into beta sheets is sort of our secondary structure. And then tertiary structure is the overall complete fold of the protein, the native fold, it's overall tertiary. I don't know, any of those words might describe its complete structure. And what's being shown here is, you know, in this one, there's some beta sheet regions, there's some alpha helices regions. Ooh. Someone sent me a really nice video last time, but it was like way too late in the semester to be relevant. I need to go find it. It was really nice, and it talked about sort of the energetics governing whether a helix would like, interact with a beta sheet and why or why not. And I think it's a good reference. I might put it on the Canvas announcement. Okay. But what we have going on here, right, so for tertiary structure, once we actually get our complete folding together, this is when things like an ion pair might form between our charged side groups, our disulfide covalent bond. I don't know what this one is. Does anyone know? I don't remember. But one of our non-polar amino acids, I have none of them memorized. I think this is valine. Valine-valine interaction, right? This is another figure that talks about that, gives more definition to primary, secondary, tertiary. Just to give another sort of key number for you here, a secondary structure requires about 5 to 40 residues to form a helix. So you could have a helix out of like 6, 7, 8 maybe amino acids, or you could have one that's a little bit longer, closer to this 40 or 50 numbers. So sort of that range over which you might have a helix would be probably useful to keep in mind, since these things can be very long, thousands, tens of thousands of amino acids long. And this is just another visual representation you might see. These are the linker structures. Usually this barrel shape here will be used to indicate alpha helices in the book. Does anyone know what this protein is? Myoglobin, right? Myoglobin. It has a heme group It has the site to, the heme group has a site to attach oxygen, but it's only one unit. So it's myoglobin. Hemoglobin it's a lot like myoglobin, but obviously has 4 subunits, 2 alpha, 2 beta. So what do we call the type of structure after tertiary that hemoglobin possesses? Quaternary, I don't know, it's a word I don't like saying, right? It doesn't roll off the tongue. Quaternary structure, 4 with a prime after it, whatever you like, right? This 4th level of structure, okay? Many proteins do have functions all by themselves. Myoglobin is an example, okay? It's just one unit and it has its own function, but they also, of course, complex with other proteins. And so this quaternary structure is when protein complexes kind of come together. We have separate polypeptide chains or protein chains and then associate specifically. Okay? And the interactions between them, though, are no longer covalent. These are our non-covalent interactions, right? Hemoglobin is one of our examples, or RNA polymerase. Again, for the purpose of, this is showing the binding site for DNA to synthesize a messenger RNA. Okay? It's obviously a big complex of many different subunits. Oh, I did put a review question slide in here. All right, talk to someone next to you about these 3 questions for a couple minutes, okay? At what level of protein structure would an alpha helix dipole be important? What might be the function of an unfolded short peptide sequence in the body, something less than 50 residues long? What would be the role of the unstructured peptides and unstructured regions within a folded protein? So talk about those for a couple seconds.